Hello grade 11. This lesson we're going to be talking about muscle fibers and how they actually contract and this is going to help you with your task for this week, which is to create your stop motion animation. So we're aware of this outline of a muscle fiber, okay? You should be able to label these things, the perimysium, the epimysium, okay? And then we have these myofibrils, the muscle fibers. We know that when a muscle contracts, it gets shorter, but in this lesson we're going to be talking about exactly how that happens. Alright, so here we've got the striated muscle cell. Skeletal muscle is striated, as we know. It's got dark and light bands. These are what we're drawing within the sarcomere, so we should get a bit of sense now of what those striations actually are, okay? A new word perhaps might be the sarcolemma, all right, which is the membrane that holds all of the muscle cells together, all right, and then within the sarcolemma we have this sarcoplasmic reticulum, all right, which is an organelle that sits within and delivers calcium in particular to different parts of the myofibril. All right, so the myofibril is down here. It's a basic contractile unit within a muscle cell. All right, this should look a little bit like the diagram that you drew yesterday. So we have this actin, all right? This is a certain type of protein. It's the thinner of the two fibers, all right? And then we have myosin, and you can see that they overlap, all right? This is actually an electron micrograph, so you can see what it actually looks like. This is not contracted. This muscle is not contracted, all right? So down here you can see the actin, okay, and then you can see the myosin, which is in the middle, all right. This dark band corresponds here to where there is no actin when it's relaxed, okay, and then the light band is over here, where there is only actin. There is no myosin in the light band, all right. That's why it's lighter in color, and this dark band is where the myosin sits. Something to note, and this will be relevant a bit later, the myosin, as well as being a fibre, it has these attachments off the fibre that's called a myosin head, and that very much helps with the contraction. Okay, so we're just going to watch this video. I want you to pay really particular attention to what happens to the Z lines, okay, what happens to the I band, the A band, and the H zone. So when the muscle contracts, what happens to each of those things?
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomere shortens. In the fully contracted muscle, the ends of the actin myofilaments overlap, the H zones disappear, and the I band becomes very narrow.
So paying attention to what happens to each of the H zones and the I band, and look also at the A band, all right? That remains unchanged. I hope you noticed there is kind of a funny little movement happening here, all right? So you could see these myosin heads were kind of attaching to the actin and moving themselves along. Imagine someone doing like the caterpillar dance move along the floor, okay? So the myosin attaches to the actin and it moves the actin forwards, all right? So it keeps doing that, and we're going to look at how that happens in the next little video. So this video is really zoomed in. You're going to see that this is the myosin and this is the actin. Okay, this is the myosin head, and then there's a place on the actin called the binding site. All right, so we're just going to see what happens between the myosin and actin.
Myofilaments are composed of strings of the proteins actin and myosin. An actin filament has two strands of actin molecules wrapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
So again, you can see it's that walking along, it's stepping along and kind of pulling the actin towards the center. That's obviously happening if you've got both ends of the myosin, it's happening on both ends at the same time. So this just shows a little bit more information about those muscle contractions. So an action potential that has traveled along the motor neuron. Okay, it's got to the motor end plate, it's got to the neuromuscular junction, and it releases calcium ions. All right, so these calcium ions, if you want to write it shorthand, it's Ca2+. That 2 should actually be superscript. It would be better if it was. All right, and that calcium opens up the binding sites. So we saw those binding sites in the previous little video. All right, the calcium ions open them up. Without the calcium there would not be any place for the cross bridge to form. All right, so that myosin head would have nowhere to join. The myosin heads are bound to these actin binding sites, making a cross bridge, and they're stimulated to move. That causes the actin to slide over the myosin towards the center of the sarcomere. Okay, ATP is then used, all right, so that's our energy currency. It's used to release the myosin head and it resets it. Then it forms a new cross bridge with different actin binding sites and continues a contraction. So putting things together, because ATP is needed for this, you would expect to see a lot of mitochondria. All right, so a lot of mitochondria in muscle cells because there's a big requirement for energy. So this happens about 5 times per second, and it ends when there's no more calcium being released. So when that nerve impulse is finished, all right, that means that no calcium will be released, so it means that the muscle will not contract. There are 2 more chemicals that we need to know the names of and know what they do, and that's these 2. We've got tropomyosin and troponin. Okay, tropomyosin and troponin. Now, you have your actin fiber here, okay, which is represented by these small circles. And on each of the actin molecules, there is a binding site. Okay, so potentially the myosin could bind to this binding site for the actin. However, when it's relaxed, it's being covered by something. All right, so it's being covered at the moment by this strange little fiber, all right, that wraps around all of the actin. And when that is there, it means, when it's there, it means that there's no way that the myosin is going to be able to bind. All right, and that fiber is called tropomyosin. Bit confusing because it's something that's on actin, yet its name is tropomyosin. Okay, so it's wrapped around the actin molecule, and when it's relaxed, there is no way for the myosin to bind. Okay, we also have these complexes called troponin, and at relaxation, they're not really doing anything at all. They really kick into play when calcium is there. So after a nerve impulse, okay, after acetylcholine, the neurotransmitter is being released, calcium ions are going to diffuse from the sarcolemma. And the calcium ions are going to have a role, and they bind to the troponin. Once the troponin has been bound to, okay, it then moves the tropomyosin out of the way, so the binding sites are exposed. So on top of instead of just saying, well, the calcium is there and the calcium exposes the binding sites, all right, a little bit more detailed would be to say that the calcium binds to troponin, all right, it binds to troponin, and that moves away the tropomyosin, which releases the binding sites or exposes the binding sites so that the muscle can contract. The myosin heads will then attach to these binding sites, and the actin will be moved in towards the center of the myosin. So you have been tasked with creating a stop motion animation, okay? And what do you need to include? Well, your stop motion animation should really answer this question. So you should be able to explain how skeletal muscle contracts, okay? It's going to be a diagram, but all of the steps that you see on this mark scheme, all right, I would expect you to have within your stop motion animation. The only two things, oh no, it is here, sorry. I was going to say troponin is not here, but it is. All right, so this kind of gives you a step-by-step model answer, I suppose, of what happens in a muscle contraction in skeletal muscle. You need to turn that into an animation so that somebody else would understand the same thing. Okay?